The overall goal of this procedure is to measure visual evoked potentials on a mouse scalp using a dry, non-invasive, and multi-channel EEG sensor.
This method can help answer key questions in preclinical translational research field to bridge the signal gap in non-invasive EEG research between human and laboratory animals. The main advantage of this technique is its non-invasiveness, which enhances comprehension of animal EEG. Furthermore, it provides safe and convenient experimental environments.
Begin by preparing 16 pins for one non-invasive electrode. Cut 2 pieces of glass fiber substrates with the size of 15 mm by 17 mm. Next, make 16 1.2 mm diameter holes using a precision engraving machine. Then, drill 16 holes into the flat substrates according to the probe coordination, evenly at an interval of 2 mm. Stack 2 substrates and apply 1 drop of fast-acting adhesive glue between the substrate layers to create a double layer of 3 mm thickness supporting 16 stable and parallel electrodes during signal acquisition. Then, manually assemble the 16 electrodes onto the substrate one by one. Solder and link each electrode's ending solder cup part to the touch-proof connector.